everyone. Thank you for attending my talk today. So I'm from Draper. We are an independent, not-for-profit R&D company based in Cambridge, Massachusetts in the United States. We are composed of mainly scientists and engineers from various subdisciplines and backgrounds. Our expertise spans many fields, from guidance and navigation systems for the Apollo space missions to MEMS and microfluidics to tissue engineering. And one unifying characteristic that we share across the board is that we are really good at making big things smaller. And so with that, we have developed Predict96, a high-throughput organ-on-a-chip platform that can be used for therapeutic screening. The device is, or each device is composed of an apical and basal channel that are separated by a microporous membrane. Cells can be grown on either side of the membrane, and each end of the channel leads to a well for media exchange and fluid flow. Within the Predict96 plate, there are 96 of these microfluidic devices arrayed in a standard 96-well format. This 96-well form factor allows for compatibility with lab equipment that is already available, such as liquid handling robots, microchannel pipettes, as well as high-content imagers. The plate is made of plastic, and so it is optically clear. We also have a pump lid that sits on top of the plate that has 192 micropumps integrated into the lid. So that's enough pumps for each apical and basal channel in every device in the platform, allowing for independent recirculating flow. So you can have the same flow rate in both channels, you can do different flow rates, and you can even turn off flow in one channel if you have cells that are particularly sensitive to fluid shear stress. And in addition, we also have integrated sensing capabilities. So we have an integrated tier system in the Predict pump. We can functionalize these micropumps so that they can double as electrodes. And with electronics integrated into the top lid, you can do automated tier readouts. We also have accompanying software where you can determine how many devices you want to read, how often you want to read them, and how many times you want to read them. And this pump is controlled by a Bluetooth-enabled controller box. So the Predict96 plate, the pump, and the controller box together make the whole Predict96 system. And so we have developed a lot of different tissue types within the Predict96 platform. Today I'll be focusing on our upper airway model, which is grown in our Predict96 ALI platform, which is just a variation of our standard platform in which we've replaced the apical channel with an open well top to allow for direct access to this culture region so that we can dry out that well and lift the tissues into air-liquid interface. So we can seed human primary upper airway cells into this apical well, lift the tissues into ALI, and culture them over the course of several weeks to generate this pseudostratified lung tissue that has all of your major cell types. So it has your columnar ciliated epithelial cells, it has goblet cells, secretory cells, and basal cells. And you can see here that structurally and morphologically, the tissue resembles what you would see in a human lung. And so there was rapid development and deployment of highly efficacious vaccines that really changed the trajectory of the COVID-19 pandemic. And there was a lot of time and resources spent on therapeutics. However, that was still a very challenging issue to tackle. It was really hard to develop novel compounds, and there were lots of candidates that were identified as potential repurposed drugs for SARS-CoV-2 that were tested in clinical trials, but ultimately showed no efficacy or little efficacy. So there is definitely a need for discovery and screening models that are predictive of clinical efficacy to address this issue. And so ideally, we would have models that are human-based. So everyone here knows that cell lines are not necessarily representative of human physiology, even if they are very high throughput. Ideally, we would have models that could go into high containment so that you could use authentic live SARS-CoV-2 virus so that you can truly look at the whole viral replication cycle as opposed to only looking at viral entry, which is what you would get with pseudotype. And we would like models that can robustly and reproducibly demonstrate SARS-CoV-2 infection, which we've heard from various groups and consortiums is a major challenge that many groups face within the MPS community. And so the Predict96 ALI lung model checks off all these boxes. We can grow the tissues and mature them at BSL-2, transport them into high containment at BSL-3, infect with various strains of SARS-CoV-2, and monitor their infection kinetics over time. So in this graph on the left, we have a single donor infected with 3 different strains of SARS-CoV-2. We infected with Washington, which was the original variant from early 2020, and then also Delta and Omicron variant. We can infect for 2 hours on the first day, and then remove the inoculum and monitor the tissue over the course of 6 days. And you can see that we get 3 to 4 log increase in viral replication in viral RNA over time, depending on what strain you're looking at. And we also see a differential response in the infection kinetics between the 3 variants. So in addition to looking at one donor against multiple strains, we can also look at multiple donors against one strain and also see some differential responses in the infection kinetics between donors, demonstrating a donor and variant agnostic human upper airway model of SARS-CoV-2 replication. So in addition to just looking at viral RNA, we can also get a lot of other different data points from a single device. So we can look at live replicating virus. We can collect media to look at cytokines and biomarker release. We can do IF to look at spatial morphological data. So this image up here, we have the SARS-CoV-2 nucleocapsid stained in red, and you can see that a lot of our columnar epithelial ciliated cells are the ones that are positive for the SARS-CoV-2 infection. And we can also look at cellular toxicity and transcriptomics. And in addition, we get highly repeatable results between experiments. So our variability is low in between devices on the same plate, as well as in between conditions on different plates. And so with that, we can do antiviral screening and studies. And so the high throughput nature of the PREDICT-96 plate allows for multiple compounds at multiple concentrations to be tested on a single plate, and also you still have space for your relevant controls. And so in this study, we tested several compounds. We tested 3 of the, at the time, FDA EUA approved compounds. So nirmatrelvir, which is Pfizer's compound, remdesivir from Gilead, and molnupiravir from Merck. We also tested the precursor to nirmatrelvir, as well as a compound that was identified by in vitro studies to be a potential drug repurposing candidate that ultimately did not make it to clinical trials. So if we focus here on the top left graph, the dark gray and black lines at the top here are our controls. So these devices were treated with virus but with no drug. And you can see that over the course of 6 days, we get about a 4 log increase in the viral RNA, which translates to about an 8,000-fold increase. And then the blue, green, and red lines are different concentrations of the drug. And you can see that by the time we hit this 1 micromolar compound, or 1 micromolar dose in green, that we have blunted viral replication. And so you can also see differential response between the other compounds as well. I think the computer is stuck. Oh, okay. Yeah, and so if we focus on the 3 FDA EUA approved drugs and look at our viral RNA copy data and compared to clinical outcomes, we can see that our data lines up pretty well with the reported outcomes in literature. And so in addition to looking at viral RNA copies, we can also image, so we can stain those same devices for the SARS-CoV-2 nucleocapsid and look at the effects of the various compounds at the various concentrations on the live virus in the cells. And we can generate a data set that complements our viral genome quantification. So in addition to testing the approved drugs, we also tested amidiaquin, which was identified by both in vitro and preclinical animal models as being a potential drug repurposing candidate against SARS-CoV-2. And it went through phase 2 clinical trials, but ultimately did not show any efficacy over the standard of care with a fever reducer or pain reliever medication. And so we infected some tissues with SARS-CoV-2, the Omicron variant, and tested several compounds, including the 2 FDA-approved drugs, nirmatrelvir and remdesivir, as shown earlier, and then also tested amidiaquin. And we can see that nirmatrelvir and remdesivir were still efficacious against the new variant Omicron, but amidiaquin showed no efficacy at any concentration, demonstrating an in vitro, in vivo correlation of our upper airway platform. Sorry, I also forgot to mention earlier how we actually do this. So when we add the drug, we still do the infection, but then afterwards we remove the virus off the top of the tissue, and then we dose the drug into the basal channel with the recirculating media, and then we replace it every day. So this is a therapeutic treatment as opposed to a prophylactic treatment. And none of this work would have been possible without our ability to work in high containment with our dedicated and experienced staff who have decades of experience between them in BSL-3. This has all enabled us to demonstrate a robust and high throughput viral replication of authentic SARS-CoV-2 in an upper airway MPS model. We have demonstrated this with 12 donors so far, but there's no reason why we couldn't do it with more. Each donor has been successfully infected on their first pass. Same with variants. We've worked with 3 variants so far, but we could work with more. And so today I spent most of my talk talking about our upper airway model. However, we've developed a lot of other tissues within our Predict96 platform. We also have other technologies in the platform, including multi-organ systems, and we are currently looking at the role of immune cells in SARS-CoV-2 infection in our upper airway lung model. And I would like to just give a shout out to my amazing colleagues for their dedication and passion to addressing these important issues. None of this work would have been possible without them. Thank you.
Thank you very much for this nice talk. We have time for questions.
Very nice talk. I'm just wondering, when you are doing the live microscopy, do you have the fluidics on, so you are still running a flow when you are doing the live microscopy?
No, you have to take the pump off.
Okay, so it removes
yeah.
the risk of having pressurized tubing in the microscope.
Yeah. Also, there's not that much space between the microscope and your plate. Yeah.
Okay, cool. And another question, if I may. How easy is it if you want to put cells on the vasolateral side of the chips?
It's very easy. You just need to load the plate and then you flip it over and then the cells will settle. So you might have to play around with densities a little bit, but we've done it on a lot of other models. So it's something that we regularly do. We just haven't done it in the lung model.
Cool, great. Thanks.
Mm-hmm.
Yeah, it's nice that you have this 96 well-played compatibility, and I understand you have both an airway as well as a deep lung model. Mm-hmm. 2 questions. Have you also, that would enable you to do so, compare the viral effects on the deep lung versus the upper airways by comparing the 2 systems? And the other one, I wonder to what extent is it possible to also do a meta deposition of aerosols, aerosolized drugs on the air-liquid interface of your system?
So yes, we do have an upper and lower airway system. We've focused most of our work on the upper airway, but yes, it would be very interesting to compare the 2. I think there were various posters here that were comparing the 2 and did see differences. That would be interesting. Sorry, what was your second question?
Deposition of aerosols.
Yes. Yeah, so we can deposit aerosols if you wanted to deposit to specific devices so that you're not, just depositing on a whole 96 well. We would need to develop some sort of isolating infrastructure, but we can definitely do that, and it's something that we are interested in doing.
Hi. I might have missed this at the beginning, but does the model that you presented have any immune cell components, or is that still the future work that you mentioned at the end?
Yeah, so I didn't present any of our immune cell work, but we do have immune cells integrated in, and it's something that we are actively working on right now.
Okay, cool. Yeah. Thank you.
Other questions?
Very nice talk. Do you do the tear measurements or barrier permeability assay in these models?
Yes, I didn't have any of the data presented here, but we are able to measure tear. We normally measure tear once a week. The tissues are dry on the top, and so and you don't want to submerge them too much because it's like drowning. So we don't want to stress the tissues out too much. So we normally measure tear once a week.
Okay, thank you.
Just in regards to what you just said about exposing the tissue to a stress by putting the liquid in, have you actually observed any increase in the inflammatory markers or maybe a decrease in tear when you keep the epithelial side and the liquid for a prolonged period of time?
So we haven't looked at cytokines specifically in the response to washes, but we have submerged the tissues for multiple days for a different effort and seen that sometimes sections of the top layer of the tissue come off. So you still have a basal layer, but maybe some of your excess tissue on the top kind of sloughs off, which I don't know if that's good or not.
How soon would that happen usually?
We see it happen maybe within a day or two of like complete submersion. Yeah.
Okay, thank you.
We still have time, yeah?
Thank you. To improve the validity of the predictive value of your model, did you try other compounds that also failed in clinical trials? Because with the COVID-19 pandemic, I think the list is quite long. So it will be interesting to evaluate more than just one, because, for example, the hydroxychloroquine works in this system, but it didn't work in clinical trials. So, yeah, I think it's probably very important to really push the system to really evaluate the predictive value. So, what's your opinion on that?
Yeah, so yes, we tested amidiaquine as just an initial pass. We didn't want to dive too deep into it on our first pass and saw this data. And so we are we do have plans to test more compounds and have tested more compounds. I just don't have that data in here. It's like hot. It's like coming. We just ran the experiments very recently.
I had one question. You said the whole plate is made of plastic. Is the bottom surface made of plastic as well?
Yeah, so the grid on the top is made of COP, and then our microfluidic layers are made of polycarbonate.
Do you think that could sometimes interfere with imaging?
No, the plate is optically clear, so we can image it. We can image up to 40x immersion if you have a long working distance objective.
And one last question. With your tear measurements, can you do them over time? Are the electrodes always immersed?
Yes.
So you can do it
continuously? So there's a software you can tell it that you want to read once every hour over indefinitely, and then it'll just run.
Great. Thank you so much.
Thank you.